Hi, my name is Minh. I'm a summer scholar from SOURCE program at UD, working at the Clean Silver Lab. This summer, I aided an investigation on fetal alcohol spectrum disorders, or FASD. It's one of the most common disability in the US, affecting 2 to 5% of the infants annually. Mothers drink alcohol during pregnancy and it leads to multiple health, cognitive, and behavioral defects in the baby. It is preventable, but the consequences last for life, and we want to explore intervention that could help these children. White matter tracts connecting brain regions are particularly vulnerable to prenatal alcohol exposure. At the cellular level, the progenitor cell differentiates to precursor cell, OPC, and finally oligodendrocytes, or OL. But prenatal alcohol exposure would delay this process significantly throughout adolescence. Exercise, on the other hand, has an opposite impact to white matter, hence making it attractive as a possible intervention method. So we aim to find out how does exercise in adolescence impact the oligodendrocyte development in the FASD brain. A cohort of 50 males and 50 females were fed with either alcohol mixed with milk or nothing. And when they grew up, they were divided into standard cages or cages with running wheel in a group of 2, 3, 4, 12 days. Then they were euthanized and brain tissues were collected. We looked into the corpus callosum, the largest white matter tract in the brain. Three types of stains were used: Hirsch in blue to mark cell nuclei, CC1 in red for OL, and anti-PTGFRA in green for OPC. And then we ran a program that randomly placed a grid, and we counted how many blue specks are in there. So OL are blue specks wrapped in red, OPC wrapped in green, and in this preliminary data, we only included data from 22 female pups. In figure 5, we didn't see exercise impacts OPC population in any group, implying its influence in later stages of OL differentiation. In figure 6, we can only see the control group being responsive to intervention, while alcohol-exposed rats didn't see any change. For the controls, we think that myelination in adolescents was accelerated by exercise. And for the alcohol-exposed, there are two possible scenarios in this case: epigenetic changes to specific regions that alter stem cell migration and differentiation, or adolescents' brain growth spurt lag leading to less potent OL generation by exercise. And after this summer, we will analyze the rest of our data with the male cohort after looking into quantity, and we want to look into the quality of myelination by investigating myelin pathology in the FASD brain. We can also add an adult time point or combine exercise with another learning assay to provide a bigger picture of exercise-induced myelination. I would like to thank Dr. Klinsova and Katrina for their incredible guidance, patience, and great care throughout my internship. I am grateful for all of those supportive friends in the Clean Silver Lab, and finally, I am thankful for the opportunity at the UD Graduate School and Dr. McAfee's works behind the scenes to make this possible.